high as Rn determines it to be, right? It's just multiplying that by 1. So if by having this Kn minus K in there adds carrying capacity, how can you phrase that in terms of interest in the competition? What does Kn minus K mean in that case?
Would it be a different species effect carrying capacity?
Well, that's what this is, and that's where I want to get you guys, but you're being pretty quiet, so I'm trying to walk you up to it. So if we just think about species 1, what is K minus N over K to species 1? As N starts out really low, this is as high as it's going to be, given the population size. As N approaches K, this goes to 0. So what's this doing? It's leveling out the curve. It's slowing the rate of increase of the population. This Kn minus N over K is the degree to which a species limits its own growth as the population increases towards carrying capacity. So what's alpha N2 doing?
That would be different species affecting.
This is the other species limiting the growth of the first one. Right. Okay, so what I wanted you guys to do for homework
Species than the
was to work out an example like this. Now I gave you 1.2. Why is 1.2 kind of a confusing coefficient to give you there? What's K1 over alpha going to end up being? You're going to calculate 10,000 divided by 1.2 after class yesterday.
It's 8,333.33.
Yeah, it's about 8,000, a little over 8,000. And then the other one I gave you was beta was the same, right? A little greater than 1. What was 12,000 divided by 1.2? Yeah, whoops. So that makes it the same as this, which is a little bit, it's very unlikely and not super intuitive. So since we're all here together, let's do that same exercise, but I'm going to increase the strength of the effect of each species on the other. So now we've got these really large values. So what do you get for these two values?
The first value you get 6,666.
Yep, good. And someone else, can you give me the other one? Yeah. Say again?
8,000.
Yeah, 8,000. Good. So you've got this for the notes, but let's try drawing it. You guys remember how to draw the isoclines? If we think about species 1, one of the equilibria we calculated. When species 2 is in a population of 0, what's the first point I should draw to draw the isocline for species 1? You guys were on a roll doing this stuff last time. Can you recall it?
Draw what K1 is.
Yeah, good. Both K units. Oh, yeah, we got. Good. And what's the other end of this isocline? Oh, let's see it here. Trevor already told us. What is it mathematically?
K2 over alpha.
It's this value. It's the population size of species 2, but it's not K2 over alpha. It's K1 over alpha. So I have it right here. Let's do it that way. So there's that zero net growth isocline. Remember that all possible combinations of both species for which the growth in species 1 will be 0. Yeah.
Why is the N one there at 10,000?
This is the carrying capacity for species 1 if species 2 is at 0.
Got it. Got it.
So red is K1 divided by alpha,
Yes. Yep.
which is 6 point.
In thousands, right?
Yeah.
That's the one.
And the y-axis is.
Yeah, what's the y-axis here? You guys remember? I had put my finger on the board and we moved it around and I asked you what it meant. What does this point right here mean in terms of both species?
There's not enough.
Low population of who?
So,
Of both, right? So I move this way, which species is increasing in population size? And species 2 is, it's staying the same because I'm staying horizontally on the y-axis, right? And if I go up, now what's happening? And species 1 is, yeah, good. So this is just population size of species 2. Does that help?
Yeah.
Okay. All right, now what about the other isocline? We gotta put our second species in there. Find out where its growth is gonna be 0. What do we start with on this axis? K2, its carrying capacity, right, which was 12, I left it up there, right? And what's the other end of its isocline gonna be? 8,000, yeah. So we get this. Now, what did we do once we had the 4 regions defined by the lines? You guys remember the vectors? Yeah. Let's start with the easy one up here. What's going to happen to species 1 and species 2 down here in this scenario? Where both are low.
It's going to increase.
Yeah. So let's put an arrow for blue and an arrow for red. And we can combine these because they're vectors. And it increases. What about up in this region where both species have very high populations? Towards the isocline, right? Towards the isocline that we talked about last time. Now, I hope you're keeping track of which isocline is which, or can interpret my little labels here. What happens in this region? What's going to happen to species 1 in this region?
increase.
Say it again?
Increase.
It's going to increase because we got to go towards that isocline, right? And what's happening to species 2?
It stays the same.
I'm not on the line. I'm off the
Increase
line.
just a little bit.
Decrease a little bit. You guys, everyone okay with that? You have to go towards the isocline for the species that I'm asking about. And it's below. Sam, did you have a question or a thought? Okay, I thought I saw it. What about up in this region? What's going to happen to species 1? The blue one. Yeah, it's got to go towards the line, right? And species 2, which way do you go to get towards the isocline? Up. So in this case, we still have an intersection, we still have an equilibrium point where both species are neither increasing or decreasing. But notice that if you deviate a little bit off of that into either this triangle or into this region here, what's going to happen?
One species will be more dominant than the other.
Yeah, one species is going to continue to increase and the other one is going to go to extinction. So that's what we were seeing here, right? Which one am I looking at? Yeah, this one. Notice that the arrows in these fields, sorry, take us away from that intersection point and towards either species 1 having a population size at carrying capacity and species 2 being at extinction, or if you end up in this triangle, species 2 increases the carrying capacity and species 1 is at 0. So this is an unstable equilibrium. How was the stable equilibrium on Monday different from this? What was different about the vectors on this field?
They were all pointing towards the center.
They were all pointing towards the center. If you reverse, if you make these coefficients smaller than 1, it's going to switch the order of the lines back to what we saw. Oh, I drew it on the board. I don't have the slide. We did it all on the board. So make sure that you go back and look at your notes from yesterday, or sorry, on Wednesday, and compare them with this, so you understand the difference that happens when you have an alpha and beta value greater than 1 versus less than 1. Your carrying capacities are pretty similar, around 10 and 12 are not that far apart. Now, the other thing we talked about was what happens if you change the strength of intraspecific competition for one of them, such that one of them has a much smaller carrying capacity than the other. This carrying capacity is much lower, half, a little more than half. I've also returned our alpha and beta coefficients back to less than 1, and I've made them asymmetrical, because they don't have to be the same. It doesn't have to be 0.5 and 0.5, or 0.2 and 0.2, or anything like that. So let me ask you about this slide, and that should wrap us up, I think. Which zero net growth isocline here is which? Can you tell which one belongs to which species? Nope, that should be a 4. Sorry. Where's my mouse? Playing with the numbers there. Now, can you tell which one belongs to which?
Is N1 the top one with K1?
Yep, this should be K1. Yeah, exactly, because it's on that N1 axis.
K2 is with N2.
Yeah. Good. 4,000 here. So what's this value going to be?
8. No, 15.
Yeah, 15,000. K1 over alpha is the other end. And this value down here, the other value, equilibrium value for species 2?
8,000.
8,000. Good. So what happens in this scenario with the vectors? What's going to happen to species 1 if you start down there? Yeah, it's going to increase towards, and species 2. And if you're way out here, species 2 is going to decrease, and species 1 is also going to decrease. But now we have a third middle region instead of 4. So what's going to happen to species 1 in that middle region? It's going to increase towards its isocline. And species 2, what's it going to do? Decrease towards its isocline. Notice the combination. So in this case, what's going to happen if you start out in this scenario, both are going to increase, and eventually they'll cross this isocline, and eventually species 1 will win and drive species 2 to extinction. In this scenario, because the strength of intra-specific competition is much lower for species 1, it's very easily able to outcompete species 2, which is already struggling from high intraspecific competition. Then the interaction of the second species drives it to extinction. Whereas if species 1 wasn't there, species 2 would grow to its carrying capacity and be okay. But here with that competition, it's going to lose out. Questions about that? Yeah.
Wait, so you said that the alpha is, that 0.75 is low?
It's lower than 1.
Lower than 1?
So that means that an individual of species 2 isn't adding one individual of species 2 isn't equivalent to adding an individual 1. But it's about like 3/4 of one, right? It overlaps. If we go back to our buffet analogy, you guys like some of the same dishes at the buffet, but not all the same dishes. So there's a few things that you can differentiate.
So the 0.5 is...
It's an even lower coefficient of intraspecific competition than this. But the point is it's less than 1. There's some differentiation. And I think up here, it's a note that alpha and beta are asymmetric. They don't have to be the same for any pair of species. What's a good way to say that? So if we think about our zebra and wildebeest. There's a lot of plant diversity on the savanna here. And the zebras are probably going to prefer the grass, maybe the wildebeest eat more of the forbs or meadow herbs that are there. So having an individual wildebeest doesn't affect the zebras the same way as having another zebra does.
Can you go back to the zero net growth isocline graph? The one you were just looking at that she had a question about. There's another way of thinking about this. You can ask yourself two questions. To what degree does species 1 limit its own growth based on its carrying capacity? Is it limiting its own growth a lot or a little? Compared to species 2, for example. A little, yeah, because it has a really high carrying capacity, which means that it doesn't limit its own growth very much. To what degree does species 2 limit species 1's growth relative to the degree to which species 1 limits species 2's growth? 0.75 versus 0.5. So one individual of species 2 is the equivalent of 3/4 of a member of species 1, essentially in terms of its effect on carrying capacity. So it doesn't limit itself very much, but it's also more limited by species 2. But look at species 2. Does species 2 limit itself a lot or very little? A lot more than species 1 limits its own self. Species 1 has less of an effect on species 2 than species 2 does on species 1, but you have this heavy amount of self-limitation. So species 2 is already limiting itself somewhat, and even though species 1 limits it less than species 2 limits species 1, that big difference in carrying capacity is going to pitch things towards species 1 prevailing. This is species 1 zero net growth isocline, right? Simply because it's not limiting its own growth, its own population growth very much. Oftentimes what we see in competitive interactions is the species with the highest carrying capacity oftentimes wins the competition, even in the face of competition acting on it, simply because it has a relatively low footprint on the landscape. You can pack a lot of those individuals into the space that is available because it doesn't limit its own growth.
I'm glad you said that. The one thing I want to remind you is when you're studying this, start with maybe this scenario and compare increasing your alpha and beta values and draw a baseline. Then increase your alpha and beta values towards 1 and draw the baseline and see how they change. Then you can see what happens if you either tip these over into the values greater than 1, or if you tip forward and start doing current capacity, so let's see what we can win. So draw all of these and see if you can play with the parameters. But do it one at a time. Either vary your current capacities or vary your alpha and beta and see how those baselines play.
Yeah, and
Also, description of this in your textbook. Once you read that, it's on the schedule that goes with these slides.
so running the numbers is, I think, useful and important, but possibly more useful and important is getting an intuitive feel for how the equations work. K is basically the degree to which one species limits its own population growth. Alpha and beta are the degree to which another species limits the growth of that species. And it's the relative strength of these two limitations that leads to those zero net growth isoclines either being separate and not crossing one way versus crossing the other way. And so if you can answer these questions relative to, I could do the same thing here with species 2. This would be K2. Species 1 limits species 2. That's beta. The degree to which the relative limitations are acting determines what form that graph is going to take. And generally, most of the time, species limit their own growth more than they are limited by another species' growth. And that's when these zero net growth isoclines make sense. Either one species wins or another species wins, or they tend to coexist.
That occurs when these values are less than 1.
Yes. Whenever another species limits your species more than you yourself limit that species, that's when you get this unstable equilibrium, in which case that's when alpha and beta would be greater than 1. And so there are intuitively logical outcomes of competition, and then there is an intuitively illogical outcome of competition, which is this unstable equilibrium. And the reason that it's illogical, and I like this room for this reason, when you look out at that plant community out there outside the window, what do you see? Do you just see one species of tree? No, you see all kinds of species of tree, because generally over time, species evolve to coexist with one another because they divide up the resources that are out there. By dividing up resources and saying, I'm going to use this part of the resource here, and another species says, I'm going to use this part of the resource over here, what they're doing is they're limiting the effect that each of them have on each other. That tends toward coexistence because alpha and beta tend to be below 1. So try and develop an intuitive understanding of what the equations are saying, as well as just being able to operate the numbers of it. I think that's
That's why
productive. Yeah, a good way to prepare for the exam is try and explain this to your grandmother. If your grandmother can understand it, then you know it well enough for the test. So if you have a problem, if your grandmother has a problem with you going through this with her, just send her to me and Dr. Judd, and we will straighten it all out.
And again, as you're studying these, if you feel like you need a refresher, just come see us.
All right. Last thing in this case study deals with adaptive radiation. Adaptive radiation is what you were supposed to prepare for today, but you'll actually do that over the weekend because of, well, the room that you see with a bunch of people not here for a variety of reasons. So, what are the goals for today? We have lots of ways of defining what a species is, and we're going to go through and share some of those with you. So by the end of the day, you should be able to explain what those species definitions are. By the end of the day, you should be able to explain different mechanisms by which species come into being, and be able to explain what adaptive radiation is and how adaptive radiations proceed. So if you're out in nature and you saw some salamanders in a forest, you might notice that they all look alike, and this is one population of salamanders, and they're all of the same species. And then when you found another group of salamanders, you might see that they look slightly different, and then you might call those a different population of species, simply because a population of salamanders, rather, a different species of salamander, and you might say that they're different because they look differently. But is that the best way of defining what species are? If your grandmother said, what's a good definition for what a species is, what would be the definition you would give her? Amanda, what is a species? How do you go about defining a species? Yeah. Okay, so certainly living. What else? Okay, so there's population. So maybe I'm not asking the question correctly. How would you delimit one species from another? How would you tell if you had two individuals? How would you tell whether they're members of the same species versus members of another species? Yeah. Okay. So oftentimes the only species concept you learned back in high school is the biological species concept, which is put forth by Ernst Mayr. Jesus, when? Oh, 1942. Groups of organisms that are potentially capable of interbreeding are in the same species. And this is easy and intuitive to think about. If you're capable of breeding with one another, you must be of the same species because your gametes are compatible with one another. And it doesn't say here, but you also, the result of that interbreeding has to produce viable offspring that are also capable of reproducing. We have all kinds of examples of organisms that are capable of interbreeding, but then don't produce offspring that are capable of interbreeding. Can any of you guys think of an example of that? Anybody? Trevor? What?
Horse and donkey, mule.
Not donkeys, mules.
Make a,
Yes, horse and donkey breed together to create a mule, and the mule is not capable of interbreeding. Sam, another one.
the liger.
Oh, the liger. Are they not capable of reproducing? I did not know that about ligers. I'm not a liger specialist. So we wouldn't consider those to be the same species because they produce offspring, but the offspring themselves aren't capable of reproducing. Unfortunately, this is really limiting. Are these two different species or the same species? How can you tell? Well, if you're applying the biological species concept, unless you live at Jurassic Park, you can't put these two creatures together and see if they're capable of interbreeding, can you? Okay, so how can you tell if these two creatures are different? Can't extract DNA, once again, unless you're in Jurassic Park, or unless you have very special situations where you can get DNA out of fossils, but those are pretty rare. What's that?
I was just saying peanut butter.
Okay, so you could just look at their morphology. Do they look alike or not? If they look alike, they're members of the same species. If they don't, they are not. The unfortunate thing about the morphological species concept is it doesn't apply in all scenarios. Here you have a frog, the great tree frog, and here you have another frog, your great tree frog. They look alike. This is the call of this frog. This is the call of this other frog. Wait. Now I'm confusing myself. Maybe this other one ended. These are two species. They're sister species, or they used to be thought to be sister species. One of these is diploid, one of them is tetraploid. This is a speciation event that came about through probably a mistake during meiosis when the homologous chromosomes didn't segregate during meiosis. And these things co-occur across their range. You can be in a pond and you can hear both of these frogs calling, but you can identify them as different from their call or by looking at their blood cells or by sequencing them or doing a karyotype of them, but they look virtually alike. So the morphological species concept would be actually terrible at telling these two species apart, because presumably these two species are on different evolutionary lineages. When this duplication of the genome occurred, one species went off on one lineage of tetraploid organisms, the other lineage went off on an evolutionary path of a diploid species. And as you can see, Hyla versicolor is the tetraploid, Hyla chrysoscelis is the diploid, and Hyla chrysoscelis actually gave rise to Hyla avivica. And so the evolutionary species concept deals with phylogenies, deals with trees, and it basically says if you're on an independent evolutionary lineage, you are a species independent of other species that are on other lineages, on other evolutionary paths. Guess what I'm going to say now? What's that? That doesn't work either in all cases, because then we have situations like this, where we have Aspidocylus gularis, which is a lizard that lives in the desert southwest, lives down in Texas in the Rio Grande region. And then we have Aspidocylus inornata, which also lives down in the desert southwest. And sometime in the past, they got together and interbred, is that even a word? They interbred and they produced a new species called Aspidocylus uniparens. Aspidocylus uniparens, the name means one parent. This is a type of hybridization that happens pretty often in this family of lizards, where the population, the species, is made up of an entire population of females. And they produce by, you don't have to move that, they produce by parthenogenesis. So each female has the ability to prompt her eggs to divide, but when they divide, they duplicate their genome, but they don't undergo cytokinesis. The cytoplasm doesn't divide. So they can produce new diploid offspring from their haploid gametes. And all of those offspring are female, and they produce their own offspring. And so in this case, if you were to apply the evolutionary lineage, each one of those individuals would be a species because they never exchange genes with another group of another individual. They're just a self-perpetuating line of clones. But that would be ridiculous to create a species name for every parthenogenic lizard running around the desert. So even the evolutionary species concept doesn't work very well. So there's another species concept called the recognition species concept, which basically says two species are the same species if they recognize one another. And this works for things like spiders, where we have Schizocosa ocreata, which is a small spider that lives in forests around here, Schizocosa craspes, which also lives in forests around here. They look morphologically alike, but they are behaviorally different, such that the males do this leg-waving dance to court a female, and the dances are different enough that females of one species don't recognize males of the other species if they're doing the wrong dance. And doing the correct dance is super critical, because if you do the wrong dance to the wrong female, rather than her seeing you as a person to have sex with or as a spider to have sex with, she will perceive you as food. You get killed and eaten. That's bad. It's bad. What does recognition species concept not do a good job of describing, helping us discern? What have I not shown you any species of in all of this discussion? Oh, no mammals, that's true. I haven't shown you any plants. Plants, obviously, the recognition species concept wouldn't apply to plants, because plants don't exhibit these types of behaviors that allow plants to distinguish between one another. There are reproductive incompatibility issues with plants, but plants have all the same issues that all of these animals have. And they may have even more issues, because plants are more prone to have these ploidy duplication, these chromosomal duplication events. And they tend to hybridize a lot more than animals do. Am I correct in that generalization?
My favorite one is there's a species that's derived from a species of ferns from a hybridization of two different parents. So as soon as that hybridization happens, you have a new evolutionary lineage. But members of those two species are hybridizing and producing those daughter species all the time, and the daughters reproduce each other. So the lineage is getting new genes from new hybridization events constantly.
And the wheat that your bread is made of is actually the result of a series of interspecific crosses that farmers made back in the Fertile Crescent, such that they took one diploid plant, one diploid grass, crossed it with another diploid grass, created a tetraploid grass, and then later on in history, they took that tetraploid, bred it with another diploid, and the wheat that we grow typically for bread flour is a hexaploid plant, a plant with 3 copies of the genome in it. So plants do freaky stuff.
Do they know they were doing that?
They knew that they were crossing things. They didn't know what ploidy was, because chromosomes hadn't been found yet. This was like 10,000 years ago, when at the start of agriculture in the Fertile Crescent. Bacteria are even more difficult, because bacteria move genes from one organism to another through what we call lateral gene transfer. And that turns out to be super common in bacteria. So I guess my point is, there are lots of ways in which we can delimit species, decide whether 2 organisms are members of the same species or not. The species delimiting mechanism that you use is going to vary somewhat depending upon the type of organism that you are interested in looking at. Some species can be delimited quite easily with the biological species concept, but there are plenty of them that can't. Other species that can't be delimited by that species concept can be delimited by the recognition species concept or the morphological species concept. When I was in grad school, we had to take a diagnostic exam in our first semester of PhD school, and it was a series of 8 hours of testing, and you got to choose 4 subdisciplines of biology. And the first question on the evolutionary biology exam was, what is a species? Had I just written down the species definition that is the biological species concept, they probably would have prescribed remedial coursework for me. So what they were really asking was, they were looking for you to flesh out what the possibilities are by which you can distinguish species. And I've given you, 4 or 5 here? I think the last count, there were something like 15 or 17 different ways of thinking about what a species is. These are some of the most common ones. So know what they are and know what kinds of organisms fit well within those delimiting methods and which ones don't fit well within those delimiting methods. It's not as simple as you might have thought coming out of high school. And that's actually one of the things you should be learning in college, especially in biology. What you're going to learn throughout your 4 years here is that a lot of the biology that you learned in high school isn't necessarily wrong as much as it is overly simplified and incomplete. And just get used to that. that process of discovering that the world is grayer than you thought it was.
One thing that helped me to think about these species concepts is thinking about each one of them, how do I relate to it, but then think about what criteria that concept implies that you might use to distinguish two individuals, whether it's a species or not. You have a concept that implies one or more criteria for distinguishing species on a cascade test. And then you'll see that they apply different concepts imply different criteria, and they apply differently to images.
So the reason that we want you to be familiar with species concepts and how people think about species is because we're asking you to think about this case study that is the finches in the Galapagos Islands, and they are all kind of morphologically similar in many ways, but they differ in their morphology in terms of beak size and coloration to a certain degree and things like this. We have three species that occur on Santa Cruz Island. The large beak finch, the medium beak finch, and the small beak finch, and those are delimited into different species. But within Geospiza fortis, which is this one here, the medium beak finch, this is the large beak finch and the small beak finch, within this species, there is a lot of variation, just like there's variation here and variation here. And so within that species, you can see that some members of Geospiza fortis have large beaks, some have small beaks, but most of them have these kind of average beaks. You've already seen these figures in different forms someplace else earlier in the semester. During the 70s, there was a drought. The drought killed off the plants. Seed abundance went way down, and as a result of that, the population of Darwin's finches went down. The abundance of seeds was going down at the same time. It was actually driving the decline in the finch population. But the morphology of the seeds that were available changed. Not because plants were producing different kinds of seeds, but because the pool of seeds that was there was getting eaten up by the finches. And the finches ate the easiest seeds to eat first. After those were all gone, it just left the hardest seeds. And so as a result of the finches depleting this limiting resource, you saw that over time, the average size of the finch beak went from like 9.5 up to above 10 in terms of, I don't remember if this is beak, this is beak depth, the depth of the beak. And so they evolved this ability to crush larger, harder seeds in response to this decline in the availability of easy-to-crack seeds. And you've already seen this figure also, showing the heritability of the beak trait, and also seeing this increase in both the beak size of the parents and also the increase in the beak size of the offspring. These are the babies. So this is, one of these figures was the parents, and this is the offspring each of those times. Now, selection is episodic. Selection is not always acting. And so selection might push you in a particular direction phenotypically, but then when that selection goes away, there's no longer selection pushing you that way, such that these finches evolved large beaks, but then after that drought was over, they slowly went back to kind of normal-sized beaks, average-sized beaks, because selection was no longer acting on the population. And so one of the things that you should know is that oftentimes evolution occurs, but it doesn't have any sort of long-term change on the morphology of the species. And this evolutionary change is what we refer to microevolution. And it can have effects on the morphology, or it cannot have effects on the morphology, but the degree of change is not such that you ever evolve a new species. So one of the ways of thinking of this might be to think of the species changing phenotype over time, but it never breaks into two populations, with each population going off on their own evolutionary trajectory. This is what we refer to as microevolution, evolution within a species. Finches, Geospiza fortis in 1980 is morphologically different than in 1990, but there's no branching into large beak finches and small beak finches. But of course, what your paper that you're reading for Monday is talking about is a situation where things are dividing. And so this is what we refer to as macroevolution. It's evolutionary change that results in the division of the species into two new species that are now on their own evolutionary trajectory. And so you have one species, one ancestral species giving rise to two or more descendant species. And so this would be a similar kind of thing. Things are changing over time, but there comes a point when the population diverges and one portion of the population evolves one type of morphology, the other part of the population takes on another type of phenotype. This is macroevolution, where you have an ancestral species eventually giving rise to two descendant species. So how does this work? Well, you just finished talking about competition, a situation where two species are competing for a common resource, and because they're competing, each species has a negative effect on the other species. So here we have, this is the small-beaked finch Geospiza fulliginosa on the island of Los Hermanos. This is Geospiza fortis on the island of Daphne Major. These are both two small islands where one species occurs and the other does not. So fulliginosa is by itself on Los Hermanos. Fortis is by itself on Daphne Major, but the two species co-occur on Santa Cruz and also co-occur with Magnorostris. These diamonds, or these triangles above the distribution, point to the average beak size of these finches. Average beak size of Daphne Major, average beak size of Fortis average beak size of Fortis on Daphne Major, fulliginosa on Los Hermanos, and the average beak size of all three on Santa Cruz. What do you notice about those average beak sizes? Oh, it would be more complete, more specific. So for fulliginosa, what is Daphne Major like on Los Hermanos compared to what it's like on Santa Cruz? It's relatively similar, but it's slightly higher than fulliginosa on Santa Cruz. In the case of Fortis, Fortis is considerably lower in beak morphology on Daphne Major than it is on Santa Cruz. So where these two species occur with one another, their beak sizes are further apart from one another than they would be when you look at them on separate islands. Both Fortis and fulliginosa have more intermediate sized beak sizes relative to the full range of variation on Santa Cruz, and when they are together on Santa Cruz, they have evolved further apart in terms of their beak morphology. Why might this be happening? Okay, so one of the things that might be happening is that they've evolved a separate beak morphology because they're having to divide up the available resources in such a way that they're reducing competition. I think I talked about competition at one point in the past. Think of a shoe seller who has like a full range of shoes in his store, and then somebody else opens a shoe store right across the street. Now those two shoe store owners are competing for the limited number of customers in Liberty. Let's say that these two shoe stores are downtown Liberty. So they can just choose to be in competition with one another, each of them carrying a full line of shoes. But what is more likely to have happen is one shoe seller will say, well, I'm going to specialize in dress and casual shoes, and the other one will say, well, I'm going to specialize in athletic shoes. That way they divide up the resources. One shop takes casual and dress shoes, one shop has people going there for athletic shoes, and they can maintain themselves because they've divided up, they've partitioned the available resource, which is people looking for shoes. This is what Dr. Judd referred to as niche differentiation. Where they occur by themselves, they don't have to specialize because there's no competition going on. But as soon as they find themselves in the presence of other species, they divide up that resource base, they become more specialized, and the niche that they occupy becomes different. So this niche differentiation. This gap in resource utilization, such that this species is using one set of seeds, this species is using another set of seeds, has been referred to in the past as the ghost of competition past, because we see this partitioning of resources, and oftentimes that partitioning is described as being the endpoint of competitive interactions in the past, although we don't have a time machine, so we can't go back and verify that. Any questions about this? This type of pattern from here to there is the same kind of pattern in your paper that they think is going on within Geospiza fortis on Santa Cruz, where this population is also going through its own specialization, where one section of this black histogram is specializing on smaller seeds, the other portion is specializing on larger seeds, but in the presence of a superabundance of seeds, that divergence is no longer happening because at Academy Bay, there's an abundance of food, and that food is all of the same type. In the interest of time, I'm going to skip this. Well, no, I'm not going to skip that. I'm going to skip. All right, so let's just skip the ring species. It's not that important. Two ways in which species can come about is through sympatric speciation, which is speciation where the organisms are all together and can interact and exchange genes with one another. And the other method of speciation is allopatric speciation. Allopatric speciation is easier because it involves separation of individuals by some geographic barrier to dispersal. So allopatric is when the members of the species that will eventually give rise to new species, they do that by the population becoming subdivided in a way that interbreeding is no longer possible. So sympatric speciation is a little harder to achieve. Sometimes it can happen behaviorally. This is a paper published back in 1989. They took fruit flies, put them in this apparatus, and the fruit flies could make a decision as to where they went in this apparatus. They could either go to the dark side or the light side. They could either be Darth Vader or Luke Skywalker. Once they were in the dark or the light side, they could choose to fly up or fly down. And then once they flew up or fly down, they could choose scents. They had two different scents. And so the researchers put a whole population of fruit flies into this apparatus. The fruit flies chose their habitat that they wanted to go and hang out in. Once they were there, they bred with one another. After they bred, they collected all the flies in these collection vials and put them back on this apparatus. And then the flies could choose to go light or dark, they could choose to go up or down, and they could choose to go towards ethanol or acetaldehyde, which are these fragrances, these scents. And what they found over time is when they looked at who appeared here and who appeared here, they found that these populations diverged from one another just by giving them the choice of these little microhabitats. These were the controls where there was no difference in color, there was no difference in going up or down, and there was no scent. So this would be an example of sympatric speciation just driven by behavior of individuals. Allopatric speciation is when you have this barrier to gene flow. This is the easiest way. And this happens when organisms disperse, or it can also happen when a physical barrier appears. So mountain range rises over geological time, or a river flows through an area and divides a population into two parts. So you can either get separation by organisms dispersing, or you can get separation by barriers to dispersal appearing. Example of this would be, they haven't developed into a new species yet, but back in the 1950s, I think it was, cattle egrets, which you see commonly around here, they're white birds. They hang out in cattle pastures. They're more common further south, but they're originally from Africa. They blew over in the 1950s in a hurricane, and since then they've spread throughout South America, up Central America, and into the United States. Clearly, those individuals are no longer interbreeding with the individuals back in Africa, so that would be a dispersal event that has separated those two subpopulations. Populations of mammals on one side of the Rockies versus the other side of the Rockies would be a vicariance event because the Rocky Mountains grew up and separated those two populations. So, adaptive radiation. It's a rapid diversification of species from a common ancestor. Oftentimes this happens after that common ancestor colonizes a new environment. So when we think about it from a resource competition perspective, we can think about how this competition affects speciation events. So you have colonization, and then you have exponential growth of the population. So imagine that you are on a montane island. This island has lowlands, it has highlands. We've talked about climatic variation on islands in the Galapagos. And now a new species of spider has arrived on this island. And it grows to the point that now this one species has now expanded so that you can find the spider at the top of the mountain, you can find the spider a little lower down on the mountain, you can find the spiders at low elevations and on the beach over here. Those are different environments. They differ in terms of temperature and moisture and all that stuff. And then, as a result of being in those different environments, they are then subject to adaptive change because the characteristics that you might need at the top of the mountain are different than the characteristics that you would need to survive and thrive at the bottom of the mountain. As a result, the species at the top might go on its own evolutionary pathway to become a high elevation adapted spider. The spiders down on the beach would become adapted to hotter, drier environments. And over time, these species that become locally adapted eventually evolve into new species. So obviously, you can think about from a resource perspective, what kinds of environments are going to be likely to lead to adaptive radiations. I was talking to a student earlier today about this. You would tend to see adaptive radiations occurring in places where they're more isolated, such that you don't get the constant influx of new species. They're going to be found in places where there's a lot of variation that would lead to this adaptive change, leading to a bunch of highly specialized adapted species. So adaptive radiations aren't going to occur everywhere. They're going to occur under specific conditions out there in nature. And so if you're still searching for an adaptive radiation, don't look everywhere. Look in specific places. Classic place to look for adaptive radiations is on islands in the middle of an ocean. Another place to look for adaptive radiations is in aquatic habitat, which is basically like little islands of aquatic habitat in an ocean of terrestrial environment. Another place to look at adaptive radiations is mountaintops that are cool and moist in an ocean of desert. All of these places are places where you might tend to find adaptive radiations occurring. I'll describe to you at the beginning of class on Monday an adaptive radiation that occurs in the Caribbean. It'll take about 5 minutes to go through. Instructions for the Zoom thing. You need to email me one person in your in-class group who is technically savvy enough to upload videos to YouTube. I will add that person to the YouTube channel so that they can upload it. After you've met with one another on Zoom, we want you to have recorded that interaction, and that one person, so send me that person's name so that I can get them in the system. That person will then be able to go onto our YouTube channel and upload that Zoom meeting recording. And that's how we'll be able to see what your presentations are like as well. If, after this discussion, you think you don't have something that's actually an adaptive radiation, come and see Dr. Judd and I, and we can help you find one that is. Questions? All right, you're free to go. We'll see you on Monday. Literature summary due on Monday. Hard copy in your hand.